Good afternoon. How's everybody doing? Today is pretty exciting because we're going to be talking about immunity. And this follows from what we just did last week with viruses and with infections. Today we're going to talk about innate immunity and acquired immunity. So let's go with the overview first and look at a video about the overview to host defenses. Hosts being basically humans, but sometimes just animals and many times vertebrates.
Host defenses can be divided into two categories: innate or non-specific defenses and adaptive or specific defenses. These defenses act together to protect the host against infection. The human body has many innate or non-specific defenses to prevent disease. These include the first-line defenses such as the skin and mucous membranes, plus the second-line defenses such as phagocytosis, inflammation, and the complement system. All of these defenses act against all pathogens and are not specific to a given pathogen. The third line of defense is comprised of the adaptive or specific defenses. These defenses recognize a specific pathogen and must be activated by antigen-presenting cells before they can work. These defenses include antibodies and cytotoxic cells. Together, these defenses make it very difficult for pathogens to penetrate, colonize, and cause disease.
And they do work together. So although this particular chapter is on the innate defenses, it will refer to a lot of mechanisms that will interact with the adaptive defenses that we'll pick up later in chapter 16. The first line of defense is the skin. No surprise there. Here is a scanning electron micrograph of the skin. That doesn't look like anything that, we think immediately that that person needs to put on a lot of moisturizer, okay? But that's the surface of the skin, which are dead cells, and they're very thickly layered. And because there's so many, layers of the dead cells, there are like alive cells under this, it's a really good protection. The epidermis is the top layer of skin. And in the epidermis, we find something called dendritic cells. And these are phagocytes. They phagocytize pathogens. So that's part of the first line of defense here in the epidermis. And then under this is the dermis. And there are collagen fibers under here. A lot of women who use moisturizers and various skincare products have collagen in them, but the collagen is really deep in there, and anything you put up here isn't really going to get down there. But anyhow, the collagen that's natural there, the fibers there, will help the skin be elastic and resist abrasions. So the skin there, with that first line of defense, is really important. It also has chemicals, perspiration, and sebum, the oil glands that keep the skin pliable. And it also lowers the pH to protect against many of the microbes there. The next group of defenses are going to be the mucus kinds of defenses. And here we've got the respiratory system, which is lined with mucous membranes. So the respiratory system consists of the nasal passages and the pharynx, the mouth, the tongue, down through the larynx, the trachea, the bronchus, and into the lungs. When you get down into the alveoli, those are pretty sterile and things don't usually, there's no mucus down there, but the mucus is up through in here. And so if we look at a comparison here, we can look at the skin and then mucous membranes. And the number of layers, are they tightly packed? This is interesting because the skin is many layers, some are dead, some are alive, where the mucous membrane are all alive. Mucus is only in the mucous membranes, and it's very moist, wet, and things called defenses, lysozymes, sebum, these are chemicals and enzymes that help ward off infection. And in the mucous membrane, in many parts of it, we find things called cilia. And the cilia, if we look here in the bronchia and the trachea, the cilia are going to be beating the mucus up and out of the lungs to remove the bacteria up in mucus, and you sometimes cough that up. And there are cilia in the uterine tubes, which we're not really talking about here. The uterine tubes beat down, the cilia in the uterine tubes beat down, and they are actually very useful for delivering the egg into the uterus from the sometimes they're called the fallopian tubes, and they deliver the eggs from the ovaries to the uterus. And then all of these organisms are constantly replacing their cells. There's a lot of self-sloughing, and that's another way that the protection of the first line of defense, getting rid of some of those cells. Another first line of defense are the lacrimal apparatus, and so these are the tear ducts and the tears, etc. When we look at this, you can see that there's a way to drain the tears, and the tears drain from the eye down through a duct into the nasal cavity and into the pharynx and down the throat. So it's not unusual when people are crying that they're snuffling a bit too, because all of that teary wet substance comes down. However, the lacrimal glands do produce something called a lysozyme that's in tears, and the tears with this lysozyme control bacteria because it kills it. Now the lysozymes that are secreted here don't hurt the eye, because the lysozymes degrade that peptidoglycan that's in the bacterial wall. And human eyes, well, no eyes really, have a cell wall like that, so they're harmless to human eyes, but they are lethal to the bacteria. So that's another part of the first line of defense that is critical. Something else, okay, so we have the skin, we've got mucous membranes, we have the eye and the lysozymes, and now we're talking about the microbiome. We did a little bit about this earlier, about how the microbiome are the friendly or at least not harmful bacteria that live in and on us. And so it's going to compete with the potential pathogens for nutrients, environment, prevented a lot of things. When we kill this, when we take antibiotics, we sometimes kill the microbiome in addition to the bacteria that it's, the antibiotics target. But keep in mind that bacteria are not the only pathogens. And so at that, they're, they're not eating the nutrients, there's not an environment, that's unfavorable anymore. There's like an open niche, and fungi and bacteria that have not been killed then can colonize in here and sometimes cause a secondary problem. But the microbiome in innate immunity will keep those pathogens at bay and reduce their numbers tremendously. There are some antimicrobial peptides that are in the skin, and then there are some organs that are going to secrete antimicrobials also. So let's take a look at a table now. Love tables, right? And we're going to look at various systems and what is in those systems and how they function. So here you go with the digestive system, starting with the mouth. We've got saliva, and it washes the microbes from the teeth, the gums, the palate. There's a lysozyme in there too. Then the stomach acid, once you swallow, the pH drops tremendously. The stomach is between pH 1 and 2, and that either kills or inhibits many microorganisms. Also in the stomach is gastroferrin. ferritin rather, and that sequesters iron being absorbed. And so it doesn't allow the microbes to use it, and microbes need iron. So, anything that the body does to sequester the iron and keep it away from microbes will also prevent microbes from growing. Then bile, which is produced by the liver, their intestinal secretions, then the movement, just moving everything through will prevent the bacteria from infection. And it moves it through by defecation, and then sometimes vomiting prevents food poisoning. You get really sick and you throw up. This isn't part of the natural sequence of events. The natural sequence of events starts with the mouth, ends with the anus, and then vomiting is just sort of an unpleasant side effect. When we look at the urinary system, it secretes urine. Urine also has a lysozyme in it, but the urine is also acid, has a low pH, so that's going to inhibit microorganisms. Vaginal secretion, again, acid, it inhibits microorganisms, and it sometimes kills sperm. So, when you're doing anatomy and physiology, you'll probably come into that in a different way. Also, you'll see that there are some iron-binding proteins here that's going to sequester it. Then the menstrual flow is going to cleanse it. Now, when we look at the prostate, that is, of course, in the males. These were in the females. And again, we see some iron-binding proteins that are going to sequester the iron, making it unavailable. To the cardiovascular system, the blood's flowing through. It's going to move microorganisms from the wounds. Coagulation is going to prevent entry of the pathogens. So, why doesn't lysozyme released in tears harm a person's eyes? You can either turn on your microphone or you can type into chat. Who remembers what I said about how the lysosome affects the bacteria? That's right, Matthew. The eyes don't have peptidoglycan. And if you were answering this in a quiz or on a test, you would then say, and lysozyme destroys peptidoglycan, which is present in bacteria. But the eyes don't have it, and so that's why it will not harm human eyes. Okay, so now we're on to the second line of defense. And this happens when the pathogens are going to penetrate the skin or the mucous membranes. This is when the pathogens can get inside the body, not inside as in the digestive tract. Consider the body as being like an elongated donut. And what we want to penetrate here is like the dough of the donut rather than the coatings on the outside of it. And so when we look at the second line of defense, it's composed of cells and some chemicals and processes. And many of them are contained in or originate in the blood. So let's look at the blood components. Now, these are the formed elements of blood. And there is a stem cell. Okay, so we've got an undifferentiated blood stem cell, and this is called hematopoiesis, which is a form of stem cell differentiation in the blood. So the blood stem cell can differentiate into any of three intermediate cells. One is an erythroid cell, and that makes an erythrocyte, otherwise known as a red blood cell. So that's the only thing that this stem cell is going to make, and it will make a lot of them. Then there are the myeloid stem cells, and the myeloid stem cells make all of these granulocytes that are important in, all of these cells that are important in innate immunity, not just the granulocytes, other ones. And they're important in the second line of defense. And then there's the lymphoid stem cells that will make lymphocytes, And those are for adaptive immunity, and we'll talk about those in chapter 16 in a lot more detail. So, and the platelets are for clotting, and those are also from the myeloid stem cells, okay? And the platelets are not a cell, it's a part of a cell that is useful in forming blood clots, and they're also present when there's inflammation, and we'll talk about that later too. So when we look at these together, okay, the basophil, the neutrophil, the eosinophil, the monocyte and the lymphocyte, all together we call them leukocytes. And sometimes we refer to these as white blood cells. The white blood cells are different from a red blood cell in a number of ways. One, they have a nucleus. The red blood cell loses its nucleus before it becomes activated in the body to carry oxygen around. Another thing about them is they're large. They're larger than the red blood cells. They're not nearly as numerous. They are made, on demand when they're needed. And many of them have very strange lobed nucleus. So they don't look like the typical nucleus that we see, okay? Especially these that are in the secondary line of defense. The lymphoid cell has a nucleus that we can recognize, but these don't. So that's how they're different from one another. Some of them contain really large granules, and they stain different colors. And so these are called the granulocytes. And these granulocytes are the basophils, and they stain blue with a basic dye, methylene blue. The eosinophils, and they stain red or orange with an acidic dye called iosin. And then the neutrophils. And the neutrophils will stain kind of a lilac mix with a mix of basic and acidic dyes. If we look at these in a blood smear, rather than in, the artist's rendition, you can see how they appear, and that their cytoplasm appears to be granular. They're at the same resolution here. You can see that the scale is the same for all of these. So they're roughly the same size, the basophil, the eosinophil, and the neutrophil. This talks about their relative abundance in just normal white blood cell amounts. So they're defensive blood cells, and what do? Well, the neutrophils and the eosinophils are going to phagocytize pathogens. They're going to eat them and chew them up. They're capable of something called diapedesis, and that means that they can squeeze through the capillaries and get out into the tissue surrounding the capillaries. So they can get out of the bloodstream and be effective there. The basophils don't phagocytize, but they release inflammatory chemicals. So they're going to act differently. The agranulocytes, and we're going to look at these here. These are agranulocytes because their cytoplasm doesn't look as distinct, it doesn't look granular. Their cytoplasm appears uniform. There are lymphocytes and monocytes. This is the monocyte, and it leaves the blood and matures into something called macrophages, which are phagocytic cells, and they devour foreign objects. So the monocytes are going to squeeze out of the blood, and they're going to be in different places of the body, phagocytizing invaders. The lymphocytes are involved in adaptive immunity and natural killer cells, and we're going to talk about those later in chapter 16. So clinically, looking at something called a differential white blood cell count can signal a type of disease. For example, if you have increased eosinophils, And let's go to the eosinophils. Okay. If you have an increase in eosinophils, that's going to indicate either allergies or a parasitic worm infection. If we say an increase in the leukocytes and the neutrophils, okay, neutrophils, and just leukocytes in general, but basically neutrophils, that's going to indicate a bacterial disease. And if we have an increase in the lymphocytes, that's going to indicate that there's probably a viral infection. So just by looking at this differential blood count, we are going to be able to get some kind of a idea about what's wrong. Okay. I've lost my place. Okay. There we go with the differential blood count. And I just showed you what it would do. All right. So we keep talking about phagocytosis. It's going to eat up the bacteria or the virus or the invader or something that's bad. And so, so the cells that are capable of this, we generally call phagocytes. And it's really not completely understood, but we believe that there are 6 stages of it. So let's look at this.
Phagocytosis is the ingestion of a solid, such as a microorganism or cellular debris, by a eukaryotic cell. Phagocytosis is used by some protozoans for obtaining nutrients, but this process is also used by certain cells of the immune system to fight infection. These immune system cells include macrophages and neutrophils and are collectively called phagocytes. Chemotaxis is the movement of a cell toward or away from a chemical stimulus. Phagocytes use pseudopods to move toward microorganisms or damaged cells at the site of infection, often arriving only minutes after infection. Chemotactic chemicals that attract phagocytes include microbial products, components of damaged cells, chemicals released by other white blood cells, and peptides derived from the complement system. The plasma membrane of phagocytes usually attaches to glycoproteins on the microorganism's surface. This process is called adherence. Adherence is made easier through opsonization, a process in which antibodies or complement proteins from the host coat the microbe's surface, serving as handles so that the phagocyte can more easily attach to the microorganism. The proteins that coat the microbe are called opsonins. The pseudopods fuse, forming a sac called a phagosome. The phagosome enters the cytoplasm, where it fuses with lysosomes that contain digestive enzymes and other antimicrobial substances. The fused structure is called a phagolysosome. Digestion of most bacteria is complete within 10 to 30 minutes. After phagocytosis, the remaining debris is eliminated from the cell by exocytosis, in which the phagolysosome fuses with the plasma membrane and expels its contents. Some of the contents of the phagolysosome may also be presented on the cell membrane through a process called antigen processing and presentation. Let's review the steps in phagocytosis. The pseudopods of a phagocyte use chemotaxis to approach the microorganism. The pseudopods adhere to opsonins that are attached to the phagocyte. The microbe is ingested within a phagosome. The phagosome fuses with a lysosome to form a phagolysosome, in which digestive enzymes break apart the microorganism. Finally, the remaining debris is eliminated by exocytosis. Parts of the microorganism are presented on the surface of the phagosome.
And we can look at the graphic depiction of all of that here with the various steps of what happens. And this is what we can observe and see what happens during phagocytosis. And some of this is enhanced by the adaptive system that we'll talk about in a little bit. Keep in mind that a key factor in immunity is going to be distinguishing self from non-self. And we're going to start looking at that here with this non-specific chemical defenses called toll-like receptors. The toll-like receptors are proteins in the membrane produced by phagocytic cells, and they bind thing called pathogen-associated molecular patterns, or PAMPs. And so this is where they are going to initiate defense responses, like secretion of inflammatory or stimulating the adaptive immune response, or even apoptosis, which is, of course, programmed suicide or cell death. So here are some of the toll-like receptors that we have experienced and found and what they do. The toll-like receptors are numbered, usually by, when they were discovered. And then the toll-like receptors respond to what? Bacterial lipopeptides, peptidoglycan, lipid A, bacterial flagella, bacterial lipoproteins, something they don't know what with influenza viruses. So these are in the cytoplasmic membrane. In the phagosome membrane, we find the toll-like receptors for various types of nucleic acids or nucleotides. There are also non-specific chemical defenses, cytoplasmic proteins that are going to bind to these, and they're going to trigger inflammation, apoptosis, or innate responses, but the mechanism is not well understood. So far, we've been talking about all of these defenses, and they're primarily against bacteria, although we did talk a little bit about viruses. We talked a little bit about parasitic worms, helminths. And now we're going to turn our attention specifically to viruses when we talk about interferons. These are protein molecules that are released by host cells, and they non-specifically inhibit the spread of viral infections. And here's how they work. The virus is going to infect the cell, and this is a double-stranded RNA virus. And you can see this is the RNA, and this is the protein coat. And the RNA goes into the nucleus, and the viral components then trigger the activation of something called type 1 interferon genes. So these are genes that are in the cell naturally, and they are turned on by these viral components. So once they get turned on, then mRNA is transcribed, and the proteins are translated, interferons, which are then released from the cells. And they bind to receptors both on the same cell and on nearby cells. So this is the infected cell. It's on the surface of the infected cell, but it binds to the receptors on a non-infected cell. And when it binds there, it activates the gene for antiviral proteins. And the antiviral proteins are inactive, but they're kind of set up. And so the interferon is kind of setting up the non-infected cells. Time passes. Time passes. Now the infected cell might die. And if you remember when we studied viruses, we talked about the lytic and the lysogenic phase. So this would be the lytic phase where the cell dies, it's releasing viral proteins. And the viral proteins are going to try and infect other cells. But when it comes to this cell that was not infected, but that had the interferon activated, then the cell, the viral components are going to activate these particular antiviral proteins. And when they do that, the antiviral proteins are going to degrade the messenger RNA, bind to the ribosomes, stop protein synthesis, and it will prevent viral replication. Now, it does prevent all protein synthesis, but eventually the viruses go away, the cell can still be alive, and once this is all taken care of, and the cell gets rid of the debris, then the ribosomes can be activated again and they can work. So this is how alpha and beta interferons work. So alpha and beta interferons are produced. Type 1 is produced in the epithelium and in leukocytes. It's induced by viruses. It stimulates the production of antiviral proteins, and a name of it is leukocyte interferon. And then the second type, beta interferon, is produced by a different kind of cell, fibroblasts. And so it does the same thing, but it's got a name of fibroblast interferon. Gamma interferon is a little different. This is secreted by activated T lymphocytes and by natural killer lymphocytes. And it's an adaptive immune response. So we're going to talk about that a little later. It will stimulate phagocytic activity in macrophages and neutrophils, and we said that these were the phagocytes. The other name for this is sometimes immune interferon or macrophage activation factor. So these are the characteristics of human interferons. The next thing we're going to talk about is complement and the complement system.
The complement system is a set of proteins that circulate in the blood serum. When activated, these proteins work together to destroy foreign cells by cytolysis, to activate the inflammatory response, and to assist in phagocytosis. The main components of the complement system are called C1 through C9.
So as you can see from this video, it's a cascade of reactions. Maybe that's where the C comes from with cascade. They're numbered by how they were discovered. And then, as you can see, once the C1 breaks up C2 into C2A and C2B, these different parts of this particular protein then can bind with other parts of other proteins, causing active enzymes. So let's look here. So there are several pathways by which complement's activated. We've got the classical pathway, an alternative pathway, and then the lectin pathway. So let's take a look at these.
Complement can be activated through 3 different pathways: the classical pathway, the alternative pathway, and the lectin pathway. The classical pathway was the first activation pathway of the complement system to be discovered. The classical pathway begins when a pair of antibodies attach to antigens, such as proteins or a large polysaccharide on the surface of a bacterium.
Okay, so right away we're talking about adaptive immunity here. Don't worry about that. Just, assume the antibodies are going to be available.
The antibodies bind and activate C1. C1 then cleaves several copies of C2 and C4. Together, the C2A and C4B fragments form an enzyme that cleaves C3, activating the complement system. The alternative pathway of complement activation was so named because it was discovered after the classical pathway. While the classical pathway requires antibodies specific to the invading microorganism, the alternative pathway requires three complement proteins called factor B, factor D, and factor P, or propertin. These proteins are attracted to microbial cell surface material, such as the lipid carbohydrate complexes of certain bacteria and fungi.
You'll notice that this is not part of the adaptive system. This is still part of the secondary um, um, defense mechanism of the innate system.
Together with C3, these proteins result in the cleavage and activation of C3 proteins, which in turn activate the rest of the complement system. The most recently discovered pathway of complement activation is the lectin pathway. When macrophages ingest material through phagocytosis, they release chemicals that stimulate the production of carbohydrate binding proteins called lectins. The lectins bind to distinctive patterns of carbohydrates on the surfaces of certain bacteria and viruses. The lectins themselves can act as opsonins for phagocytosis, but also activate C2 and C4, which leads to the activation of C3, leading to the activation of the rest of the complement pathway.
So we have three known, well-defined mechanisms, cascades, if you will, that will activate the complement and cause cellular responses. So now that we've got activation of cellular responses, what are those cellular responses?
Once the complement cascade is activated, several events occur that ultimately result in the destruction of the pathogen. C3A and C5A act as chemotactic factors, attracting phagocytes to the site of infection. C3A and C5A also bind to mast cells, triggering the release of histamine and other chemicals as part of inflammation. C3b acts as an opsonin, coating the invading cell. This coat makes it easier for the phagocyte to adhere to the invading cell during phagocytosis. C5b binds to C6 and C7, which attach to the plasma membrane of the invading cell. C8 and several C9 molecules bind to the C5b, C6, C7 complex, forming a cylinder-shaped membrane attack complex, or MAC. The MACs form circular holes in the membrane of the invading pathogen, causing water to enter the cell and ions to leave. This results in cytolysis, killing the cell.
I want to go back to that killing the cell picture. And here you can see what looks like a rod-shaped bacteria. And here you see nothing that looks like anything that's alive. It's got a big hole in it and it's not going to be able to function at all. And indeed, you can see here a transmission electron micrograph with many of these holes in it. that were produced by the complement to kill the microbe. All right, the next second line of defense that we're going to look at of the innate immune system is inflammation. Two kinds of inflammation: acute inflammation, chronic inflammation. You may have heard a lot about inflammation in all kinds of health things that you need to have anti-inflammatory fruits and vegetables, an anti-inflammatory diet, or some things cause inflammation. It's very popular to look at gluten as an inflammatory molecule, and a lot of people want to go gluten-free because they think that they feel better with that. If you have, okay, now I can't remember, what's the disease where you absolutely can't have gluten? Celiac disease. If you have celiac disease, yes, it's really dangerous, and you can't absorb food well, et cetera, because of inflammation. So let's take a look at inflammation. Inflammation is a secondary line of defense, although sometimes it goes awry, and it is not in response to a pathogen, but is a medical condition of its own. So what happens with inflammation? Let's look at the trying to find the video. There we go. I was looking at some of these videos from Pearson earlier today, and embedding them seemed to be easier than going directly to them. But I don't know whether it's Pearson or whether it's ACC or whether it's, Spectrum, or whether it's just my computer. If this doesn't seem to be working well. I'm trying to find it in another source. I'm going to the study area in Pearson. And, well, let me just show you what I'm doing. In the study area of Pearson, there's a place for animation and videos. And I'm not sure if it's here or in the microbioflux. Not here. Try it this way. Come on, Dr. Bowman, microbiology.
When tissue is damaged, For example, as shown here by an infected cut, one of your defensive responses is inflammation. This video examines some aspects of this defense. Inflammation is a general, non-specific response to tissue damage. Damage could result from many causes: cuts, heat, abrasions, sunburn, disease, or pathogens. There are two major types of inflammation: chronic inflammation and acute inflammation. Chronic inflammation is long-lasting, damaging, and can be life-threatening. In contrast, acute inflammation develops quickly, ends quickly, is typically beneficial, and helps eliminate pathogens from the body as part of the innate immune defense. Chemical mediators trigger inflammation. These inflammatory mediators include bradykinins, released during blood clotting, prostaglandins and leukotrienes, released by macrophages, and histamines, released by platelets, basophils, and non-blood cells called mast cells. Inflammatory mediators can be released in a variety of ways. Let's examine one. Basophils and mast cells are filled with vesicles that contain inflammatory mediators. Certain proteins in the blood that are produced following infections, particularly complement proteins C3a and C5a, attach to receptors on the cytoplasmic membranes of basophils and mast cells. This causes the vesicles to release their contents. Now let's examine the effect of these inflammatory mediators on a blood vessel. The walls of blood vessels are made of cells that normally have narrow gaps between them. A small amount of fluid leaks into the surrounding tissues. Blood cells, such as these erythrocytes and this monocyte, normally remain within the blood vessels. But when inflammatory mediators attach to the blood vessels dilate. That is, they get larger in diameter. And since dilation is occurring in many blood vessels, more blood is delivered to the tissue. Blood is red and blood is warm. So more blood means more red color and more heat, two signs of inflammation. With dilation, the cells in the walls of the blood vessels move further apart, creating larger gaps between the cells, which are called interstitial spaces. This allows much more blood plasma to flow into the surrounding tissue, what is sometimes called third spacing, producing edema or swelling. Now we have the last two major characteristics of inflammation: swelling and pain. Pain results in part from compression of nerve endings in the surrounding tissue. Some leukocytes, notably neutrophils and monocytes, can squeeze through interstitial spaces, a process called diapedesis. The defensive nature of diapedesis should be obvious. Leukocytes phagocytize pathogens. In summary, chronic inflammation is long-lasting, damaging, and can be life-threatening. Acute inflammation develops quickly, ends quickly, and is part of the body's protective innate immune defense. Inflammation is characterized by four things: redness, heat, swelling, and pain. Inflammation involves the dilation of blood vessels. Dilated blood vessels deliver more blood, resulting in the redness and heat. Inflammation also results in widening of the interstitial spaces. As more fluid moves into the surrounding tissue, swelling and pain occur.
All right, so we've already gone through most of these. Okay, so these are the chemical mediators that Dr. Bowman was talking about in his video that contribute to the inflammation. The next thing, and I think is the last thing we talk about, is in the innate secondary line of defense is fever. And we define fever as a body temperature over 37 degrees Celsius, which is 98.6. It results when these things called pyrogens, which are things that will trigger the hypothalamus to increase the body's core temperature. And there are several different kinds. There can be bacterial toxins. They can be the cytoplasmic contents released when by lysis, antibody antigen complexes, and sometimes they're released by the phagocytes that have phagocytized bacteria. The exact mechanism isn't known, however. They're calling it a theoretical explanation, and this is a hypothesis. It's not a theory because it has not been well tested, but let's let it go. It's a hypothetical explanation for the production of fever in response to the infection. So here are chemicals secreted by phagocytes. So this could be the chemicals created by phagocytes or any kind of a paragen. And it travels to the brain into the hypothalamus. The hypothalamus then secretes prostaglandin, and it resets the hypothalamic thermostat. Okay, so this is part of anatomy and physiology, and we'll get a lot more of this then. Then the nerves cause shivering, high metabolic rate, inhibition of sweating, and vasoconstriction. These will increase the body temperature and to the point set by the thermostat. And it continues as long as these paragens are present. So why fever? Well, it enhances the effects of the interferons. It will inhibit the growth of some microbes, and it could enhance the activity of the phagocytes or the cells with specific immunity and the process of tissue repair. So when we look at the summary of everything that we've looked at for the innate system, we see the first line and the second line of defense, and the barriers and associated chemicals, the phagocytes, extracellular killing, and then complement. Then in the second line, we've got an increase in the resistance of cells to the viral infection, and it slows the spread of disease by the interferons. There are antimicrobial peptides, and inflammation and fever. So all of these are useful and are non-specific. They are present at birth. They are not anything that has to be developed or acquired. They're present with everyone, and they're useful in conjunction with the adaptive responses many times. So we're going to take a 10-minute break, and then when we come back, we're taking a 10-minute bio break. When we come back, we're going to go into a different collaborate session and do chapter 16. However, I can take any questions you might have now, if anybody has questions. Okay, I'll see you in the next session for chapter 16.